Welcome back, everybody. We're going to be looking at cells this week in our Concepts of Biology book. It's chapter 3, but as you see with my red circle on the chapter outline, we're going to divide this into two different sections. We'll do sections 1, 2, and 3 first this week, and next week we'll do the other 3 sections. So this week we'll be looking at cells, the difference between the main types of cells, which are prokaryotic and eukaryotic cells, and then we'll look at the complex eukaryotic cells with all of their working parts. We'll save a lot of the cell membrane and how things are transported across the membrane for next week. So sections 1, 2, and 3 for this week. So first of all, we're looking at how are cells studied in section 1. Our goals are to be able to describe the roles of cells in organisms, compare and contrast light microscopy and electron microscopy, and summarize what cell theory is. Alright, so in the picture here, I love these pictures. This is really zooming in, really getting close. This is called a transmission electron micrograph. This is actually of a picture of something that's inside of a cell. So we are going, yes, that small to see a working part inside of a cell. We need kind of an expensive microscope for that. It's called an electron microscope. So you're actually looking at what's called or nicknamed the powerhouse of the cell, the mitochondria right there. So that is one of the things that we'll be mentioning during this chapter as well. And here you actually get to see what it looks like up close and personal. Here's some other microscopy examples. So we've got some nasal sinus cells. Very nice, done with just a regular light microscope like you might see in a classroom. Here we have some onion cells, also done with a light microscope. You could use that in a regular classroom as well. And then over in picture C, that one, we're going to need that expensive microscope, the scanning electron microscope, where we're looking at some bacterial cells. That's going really close right there. So even though all of these are cells, they look very different from one another, they do share some basic characteristics. So what is a cell? It's the smallest unit that has the properties of life. It's the basic building block of all organisms that we still consider alive. As we said, cells can differ in size and shape and function, yet they would have some things in common. Each cell would have a plasma membrane, or sometimes also called a cell membrane. They have cytoplasm, which is kind of the jelly-like fluid and some of the other stuff that's in that fluid. And then if you're a eukaryotic cell, you would have DNA and a nucleus, but not all cells have a nucleus. So some just have what's called a region of DNA if you're a prokaryotic cell, but they do have that in common. Whether you're a prokaryotic or eukaryotic cell, you still have DNA, because remember, that's one of the things on our checklist of life, is that all living things on Earth use DNA as their genetic material. Some types of cells just kind of have it in the cytoplasm in a certain area, and other types of cells, like our cells, which are eukaryotic cells, actually have it packaged really nicely in a nucleus. There's single-celled organisms and multi-celled organisms, which you probably already know. A lot of single-celled organisms are prokaryotes, or what we also call bacteria, but there are some other more complex types of cells that are single-celled as well, like protists. And in fact, there's a type of fungus that has just a single cell, and that's yeast. And then there's maybe the more familiar multi-celled organisms like plants and animals and fungi, and a lot of types of protists that have cells that need to interconnect and work together to form different structures like tissues and organs, and sometimes even going as far to making organ systems as well. So with our vocabulary there, again, all cells have in common that they have a plasma membrane. cytoplasm and a region of DNA. So a plasma membrane is the cell's outermost membrane, sometimes just also called a cell membrane. It consists of a lipid bilayer, which is 2 layers of lipids called phospholipids that surround the whole cell. Remember, an important thing about lipids is that they don't dissolve in water, which is good because we don't want our cells to dissolve and just break apart. We want them to hold their structures. So having a plasma membrane made out of lipids is a really good choice for an organism who's like 70% water. And then a lot of organisms may also have organelles or little working parts inside of them, and those little working parts called organelles may also have membranes that are made out of the same type of material as the plasma membrane. So an organelle is a structure that carries out specific or specialized metabolic functions. Like in our first opening picture, I mentioned the mitochondria here. That's an example of an organelle in a eukaryotic organism. And we can see its little membrane going around the outside here. And we can even see parts here, these lines on the inside are places where the membrane kind of folds in on itself to make more surface area within this organelle. All right, then the cytoplasm is the semi-fluid substance. It's kind of jelly-like, enclosed by the plasma membrane, and would entail having other substances within it, other molecules, and if it's a eukaryotic organism, would also consist of the organelles. And then if you're a eukaryotic organism, you would have the nucleus, which it's not necessarily always in the exact middle of a cell, but it's in there somewhere. And it's a kind of what we might call the working center of the cell, and it would have its own membrane, actually 2 layers of membrane that holds the nucleus together, and even what's called the nucleoplasm on the inside of that, its own fluid and components. And if you're eukaryote, then your DNA would be in there as well. So we've got some very good vocabulary on that slide that you need to make sure you know those terms. How are cells studied? After all, cells are really small and you can't really see them with the naked eye, so we need some help. Luckily, there have been scientists and persons in the past who have developed a way to put lenses together and lights to be able to see the unseen, and those are microscopes. Those are instruments that magnify an object, and images taken with a microscope are called micrographs. Here's a little reminder of some metrics for us here. In the world of microscopy, we're often dealing with a unit of what we call micrometers or micrometers. We use the Greek symbol mu, which looks like this that I'm pointing to right there. And look how small it is, 1/1,000,000th of a meter. A typical human red blood cell is about 8/1,000,000ths of a meter, or 8 micrometers in diameter, just to give you an idea there. And the head of a pin's diameter is about 2/1000ths of a meter, and 250 red blood cells could fit on the head of a pin. So we're dealing with some really small stuff. If we're trying to look at cells, eukaryotic cells are small enough, but if we want to see prokaryotic cells, we have to go smaller yet. And if we want to see inside of cells and look at organelles in a eukaryotic cell, we have to go even smaller yet. So very small sizes, and thank goodness for being able to put some lenses together to be able to see these things. So for instance, in a regular light microscope, or sometimes also called a compound microscope or a compound light microscope, we might see this in like a regular high school classroom or a college lab. You put 2 sets of lenses together and pass light through both of them, and it magnifies the image. And so if we're passing light through our specimen, that means our specimen needs to be very small and transparent, like a smear of cells would be. And then we would be able to see that. We can see live things, like if we take some pond water and look at it through a light microscope, we can see living cells like algae and things and amoeba in there. But if we want to see some of the structures a little bit better, we might use a certain type of stain to do that. Unfortunately, that stain will probably kill the cells on that specimen slide. Something to be aware of when you're using a microscope, because you're using two lenses, it actually changes the orientation of your image. It flips it upside down and reverses it left to right. So as you're viewing things under a microscope, it's something to be aware of. And if you're moving your slide around, if you physically move it to the right under the microscope, it's going to look like it's going to the left. If I physically move my slide up under the microscope, it's going to look like I'm moving it down, and then flips things backwards. So you may recall in high school doing a microscope lab using an E and seeing how a microscope will flip that E left to right and upside down as well. Light microscopes are good for seeing animal cells and other eukaryotic cells like funguses, protists, and plant cells. However, if we want to see a prokaryotic cell, like a bacteria, that's a lot smaller than a eukaryote cell. We usually need another type of lens that some light microscopes can handle. It's called an oil immersion lens, where we put a drop of oil on top of our bacteria specimen on the slide, and then we lower that oil immersion lens down in there, get it into view, and we can magnify about 1,000 times with that to be able to see a prokaryote or bacterial cell. However, with a regular eukaryote or animal cell, we can settle for magnifying around between 100 and 400 times to be able to view those cells and study them. Some important parameters with light microscopy are your magnification. That's the degree that you enlarge your object, such as enlarging it 400 times, or with bacteria 1,000 times. And resolving power, the ability of the microscope itself to distinguish between two different points or separate two different structures. The higher the resolution of the microscope, then the closer the objects can actually be, and you can distinguish them from one another. Another type of microscope that you might see in a regular high school or college classroom would be a dissecting microscope. Notice the magnification is a lot less than a light microscope. In this case, we're not going to pass light through our specimen. We're going to look at the surface of our specimen, and we only need sometimes like maybe 10 times, maybe 20, up to about 80 times for our magnification. So some good examples that I like to use, and what I like to do with the dissecting microscope is look at my fingernails under there. When I look at my fingernail right now, you know, they look like regular fingernails, but under a dissecting microscope, you can see a lot more detail, the detail of your fingernails, the skin around them. You can even see dirt and fibers and things that get stuck onto your fingers. Looking at maybe a dead fly under a dissecting microscope, you can see the detail of the wings or the scales of a small lizard, if you can get your lizard to sit still enough under that dissecting microscope. So those are some examples there. With this type of microscope, you don't have to worry about flipping or reversing your image. So here's some pictures of those types of microscopes right here. This first one is a typical light microscope, although it gives you an extra choice whether you just want to look through one eyepiece at a time or do the binocular where you can look at through two at a time and use both eyes. Put your specimen, I see there's a slide already on the stage on that one. Light down here shining through your specimen and then passing through two lenses. And then this revolving nose piece or right here where you can choose the different magnifications that you want for your specimen. This is called the dissecting microscope over here, where you put your specimen. Imagine putting your finger here. You're going to look at your fingernail, and then you can lower or raise this right here and be able to get it into focus. So typical types of microscopes in a high school or college biology lab, definitely there. But sometimes we can go a little bit more complex and expensive and use an electron microscope. So electron microscopes use a beam of electrons instead of light, and this is going to allow for higher magnification, better resolving power. Unfortunately, this will kill your specimen. These are pretty big microscopes. They're expensive, they're bulky, and they're probably more for a more specific research lab. A scanning electron microscope, there's a few different kinds. Of this type has a beam that moves back and forth across the cell surface, and it renders the details of the cell surface by reflection. You have to cover your specimen in a substance that's made of metal like gold, but it does give you a good view of the surface of the thing that you're looking at. And then there's the transmission electron microscope. That's one of my favorites because it really does get in close to something like an organelle inside of a cell, like that mitochondria that we looked at back up here. This was used with a transmission electron microscope. So we've actually gone through the membrane and we're looking at an organelle inside of a cell right there, the mitochondria. So with this one, then you transmit your electron beam through the cell and you get good details of your internal structures. So I put that it's good for viewing cell organelles. So here's some more microscopy pictures, some Salmonella bacteria here, viewed with a light microscope. Notice when we use just a regular lab light microscope here, we're not getting much detail. We can see the bacteria are there, but not much detail about the cells themselves. So we definitely need that electron microscope like this over here, where we're seeing those same Salmonella, but up so much more closely. These are pretty cool. These are some cells that are viewed through a pap smear from a human, from a cervix of a human, the human papilloma virus. So these are actually cells, and we can see that they've been invaded by a virus. All right, let's move on to section 2, 3.2. We're going to compare the two major types of cells, prokaryotic and eukaryotic cells. We'll be able to name some examples of those by the time we're done, and some examples of eukaryotic organisms, compare and contrast the different types of cells, and describe their relative sizes. So keep going back to this picture right here has a prokaryote cell here on the one side and a much bigger eukaryotic cell. These are not to scale, but keep going back to these to visualize what these types of cells look like. But let's first take a look at cell theory. Remember, a theory is very important in science. It explains quite a large area of something, how a phenomena in nature works, and has a lot of evidence and stood the test of time. It's made of a lot of research, many hypotheses that you have not falsified, and so cell theory, germ theory, evolution theory, those are examples of theories, and they all help explain how the natural world works. So with cell theory, there's 4 generalizations. Every living organism consists of 1 or more cells, so they're unicellular or multicellular. Cell is the smallest unit that's considered alive, and you could either have just the 1 cell or part of a multi-celled organism. All living cells come from the division of pre-existing cells, and that cells use DNA if you are living on Earth. Some of the parts that they share, some of these we already know that we already talked about, the plasma membrane. And remember, that's also called a cell membrane. It separates the inside versus the outside of a cell, the interior versus exterior environment of a cell, and it's made out of the lipids so that it doesn't dissolve in all of the water that's inside of the organism. There's the cytoplasm, the jelly-like region that's between the cell membrane, and if you're a eukaryote, then in between the nucleus. The DNA, the genetic material, and then another thing, this one might be new for you, ribosomes. So ribosomes are teeny tiny little structures inside of cells. They're made out of RNA, which is a nucleic acid, remembering from our last lecture. Nucleic acid, RNA, kind of folds in and it makes this two-layered structure that to me kind of looks like two fists, and then they clump together like this, and that's what a ribosome looks like. Is like two little fists of RNA clumped together, and they're what we call the protein synthesis areas, or they basically make protein in the cells. And so that one doesn't matter if you're a eukaryote or a prokaryote cell, you have all of those, the plasma membrane, cytoplasm, DNA, and ribosomes, because proteins make up the structures and many things about any type of cell, so you've got to have those ribosomes to be able to do that. And you have to be able to code for those proteins, so we need that DNA as well to do that. So though they all have those structures, prokaryotes and eukaryotes may differ in how those structures are organized and how they look, even how some of those structures are composed. So here we go. Cells fall into two main types of categories, the prokaryote category and the eukaryote category. If your cell is in the first category, we call that a prokaryotic cell. And if your cell type falls in the second one, then you have a eukaryotic type of cell. So prokaryotic cells are single-celled organisms, not all single-celled organisms though, but single-celled organisms including different types of bacteria. And the main thing about prokaryotic cells is that they do not contain a nucleus. In fact, the Latin meaning of the word prokaryote means before the nucleus. Eukaryotic cells can be both single and multi-celled organisms, and they do have a nucleus, and that nucleus houses that precious DNA. So let's talk about prokaryotic cells first. Again, DNA in a prokaryotic cell is not found in a nucleus. Instead, it's floating in the cytoplasm in kind of a darkened central region called the nucleoid region. There's no membrane-bound organelles, like I mentioned the powerhouse of the cell, the mitochondria, earlier, which we find in eukaryote cells. They are not found in prokaryotic cells. So no membrane-bound organelles, no nucleus. And there are two basic domains of prokaryotes. They are the bacteria, sometimes also called eubacteria with the EU in front, but a lot of times I just like to say bacteria. And then the archaea or archaebacteria. So not only are these two separate kingdoms, but also two separate domains because of the differences in their cells, but they do have in common that they're single-celled, they're a type of bacteria, and they are prokaryotic types of cells with no nuclei and no organelles. Both types of these prokaryotes, though, have cell walls, although the substances that make up those cell walls are different. Cell walls are structures that surround the cell membrane or surround the plasma membrane, and they offer extra protection, extra structure, help maintain the shape of the cell, and they can even prevent dehydration of the cell as well. In eubacteria, regular bacteria, the main type of substance that makes up the cell wall is called peptidoglycan. Now there's some eukaryotes that also have cell walls. In fact, a lot of eukaryotes have cell walls, including plants and fungi and protists. But their cell wall is made out of something completely different that we'll get to eventually. Many bacteria will have a capsule made of a type of polysaccharide. Remember, that's a carbohydrate that will help attachment to something like maybe a host cell. And some also have flagella, which is a long whip-like tail that they may use for locomotion. And some may also have other hair-like structures called pili, which they can use to exchange genetic material and also use for attachment as well. Here's a typical picture of what a prokaryote looks like. This one actually has a flagella, or singular flagellum. Here's the DNA floating in the cytoplasm. That would be the nucleoid region. Then we also have lines that point to the inner cell membrane made out of the lipid bilayer, the cell wall for extra protection, and then even the capsule that's around it. So that bacteria has a lot of surrounding structures for protection there. And then the pili, which are little hair-like structures, again, that can be used for attachment and also to help the exchange of genetic material between bacteria or other prokaryotes. How about those eukaryotic cells, though? Because that's what we have. We're animals in the animal kingdom, and we have this type of cell. So animals, plants, fungi, and protists, those four kingdoms, all have in common that they have a eukaryotic cell, and because of that we are put into the domain eukarya because of our type of cell. And this means we have at least one membrane-bound nucleus or nuclei in our cells. Our muscle cells, I don't know if you know this, but some cells like our muscle cells have more than one nucleus. We also have complex membrane-bound organelles that have specialized structures, like the mitochondria, who makes energy. So why are cells so small? There's this, what's this called? It's called the surface to volume ratio, which limits our cell size. Basically, if a cell gets too big, it would take too long to diffuse nutrients and oxygen gases and things like that into the cell and to get everything that's needed all over inside of a cell. If a cell got too big, it would take too long for that and the cell would die. So cells are limited because of that. They need to be small, so it quickens the inward flow of nutrients and gases and the outflow of wastes. So that's what we mean by surface to volume ratio there. It's a relationship in which the volume of an object increases with the cube of the diameter, but surface area increases with the square of the diameter. So basically, that sounds a little complex there, but basically it means that cells need to maintain a smaller size to help the flow of materials back and forth to get what they need or to get rid of what they need to get rid of. So you want a large surface to volume ratio when you're talking cells. So sizes of cells, what are they like? I like this chart because this shows us some relative sizes on a logarithmic scale here. So let's start with what makes all matter. There's an atom right there that makes up all matter, which then can build things like macromolecules like lipids and proteins. So nanometers, right here, 1 nanometer, 10 nanometers, that's going really small there. That's, if we want to see those types of things, we need those expensive, bulky electron microscopes for those. And then even then, the images are so small. Bacteria at 100 nanometers. And then a mitochondria, which is an organelle in a eukaryotic cell, at 1 micrometer. Finally, up here we've got animal and plant cells, very similar in size. A human egg cell, notice how that's a little bit bigger. You can almost just see a human egg cell with the naked eye. It looks almost like a little pinpoint It's the largest cell. It's got to have a lot of stuff so that you can get a human going, have enough stuff to divide from one cell into 100 trillion over 9 months. So that's a really big cell. Then a frog egg, chicken egg, ostrich egg, and then finally a human. And then we have some pictures of some very well-known eukaryotic cells, including the animal cell and the plant cell. And our next goal here is to take a look at eukaryotic cells and name, visualize, and figure out what these structures do in these eukaryotic cells. So come back to this picture and visualize these often. All right, so section 3. Eukaryotic cells. Our goals are to describe the structure of eukaryotic plant and animal cells. Yes, I know there's also fungi and protists, but we'll concentrate on plant and animal cells this time around. It will state the role of the plasma membrane, though in our next week we'll get into more detail about the plasma membrane. Then summarize the functions of major cell organelles, and then also what holds the structure of a cell, the cytoskeleton and the extracellular matrix outside of the cell as well. So let's start in with the plasma membrane. The plasma membrane serves as a selectively permeable barrier. That means that it's picky about what things get in and out of it, and it separates the internal and external environments of the cell. Again, it's made out of lipids, which do not dissolve in water, which is a good thing. We don't want our cells dissolving in all the water that's inside of us. And we kind of call it a fluid mosaic. We have these 2 layers of phospholipids and then also proteins dotting within the phospholipids, and it's kind of fluid. Everything's just kind of floating in there. Some things are switching places, and so they call it the fluid mosaic model. Lipids are organized as a bilayer, that's a double layer, and if we remember from our last chapter, then there's this phosphate group at the end of each of the phospholipids, and the phosphate group gives the end of the lipid a negative charge, which makes it polar and actually makes it okay with being around water. It makes it hydrophilic. And so those ends point outward, and then they're kind of like little tails on the other side that are hydrophobic, that don't have the phosphate group. They point inward. So on my hands here, if you think of this area as the area with the phosphate group, the hydrophilic end, and then the hydrophobic end are the tails here, the 2 fatty acid chains. They point on the inside of the lipid bilayer because it's like they move inward because they don't want to be in contact with any of the water around your cells. So if they point inward towards each other, then they can be safe from all that water that's in your body. And again, they're kind of always in motion and fluid, and so they might switch places with one another. Can't do that with my hands. But they stick together because of that, well, the non-attraction for water, that the tails want to stay inward. And we'll look at some models of that in a little bit. And because of the fact that these are lipids, it makes it very picky about some of the substances that are going to get through. Now, if you are a lipid substance and nonpolar, you're probably going to be able to go through that lipid membrane and diffuse across much more easily than if you were a polar molecule, which those tails, if the polar molecule came in there, it would not like that. So some things diffuse across easily while others don't. Some cell membranes have microvilli, which are little structures that point outward, and they help to absorb material. So like a structure like your small intestine would have a lot of microvilli hanging off of their cells to be able to absorb the nutrients that are coming in through digestion. So here's our model, the fluid mosaic model. The little red heads of the phospholipids, those are hydrophilic. They love water, they're polar. The little blue tails pointing inward, those are hydrophobic. They do not like water. Those are regular fatty acid chains, so they are nonpolar. And then that kind of sticks together in that double layer or lipid bilayer. The blue round objects, those are proteins that are kind of dotting the plasma membrane itself. Some of these proteins have other things attached to them, like sugars, carbohydrates. You'll notice these proteins have different shapes. Some of them are going to act as transporters of different substances across the cell membrane. That we'll be talking about in our next section next week. And these little orange or yellowish things, those are cholesterol. Those help to make the cell membrane a little more flexible, especially if you live in a cold environment. So all of this together then makes the fluid mosaic model that makes up a cell membrane. The cytoplasm of a eukaryotic cell comprises the area between the membrane and the nucleus. And in there you're going to find the cytosol, which is the gel-like fluid. You'll find organelles if you're a eukaryotic cell, and then other structural things like the cytoskeleton made out of protein fibers, and chemical components like solutes and ions. It's made out of a lot of water, 70 to 80% water, and is the site of many, many metabolic reactions that happen within our cells. So let's look at the cytoskeleton here first. The cytoskeleton is what holds the structure of the cell, and it supports it, and also helps with the movement of the cell itself and things inside of the cell, like when chromosomes need to move and split into two and have the cell split in two. The cytoskeleton is involved with that. So the cytoskeleton is basically, it says, a dynamic network of protein filaments. Dynamic means it's ever changing. They support and organize and move the cell and the internal structures. There's three main types of protein fibers. They're the microtubules, microfilaments, and intermediate filaments, and those basically differ by size. They do have in common that they're made out of these contractile microfilaments. It's the same microfilaments that make up your very own muscle cells, myosin and actin. So those are both two types of proteins that are contractile. The centrosome is a region near the nucleus in animal cells. You don't find it in plant cells, but it consists of a bunch of those microtubule proteins, and basically it acts as an organizing center to help a cell when it's ready to divide. So the centrosome contains a pair of centrioles. They're like little rod-shaped areas that have 9 microtubules each. Right before cell division, the centrioles actually double. So then instead of having one pair of them, you have 2 pair. Then they move to opposite sides of one another, and they help set up all the other microtubules as the cytoskeleton changes during cell division. And they help to pull chromosomes apart so the cell can divide, and you get the right number of chromosomes on each side for the dividing cell. Flagella and cilia. So the flagella is more like a whip-like structure and might only be 1 or 2, whereas cilia are little hair-like structures and often cover a whole surface. So they're similar in that they're made out of these protein filaments, and they act in movement of the cell or moving something along, like in the fallopian tubes. There's a bunch of cilia there that help to sweep the egg through the actual fallopian tubes. And so they may line a surface and be involved with movement of particulate matter. So here's some pictures to help give us an idea to visualize these things, how a cytoskeleton can help support the cell like its very own skeleton, although it's made out of proteins instead of bones like we have. And these areas here look pretty neat. Those are the centrosome areas and how they branch out and how they're going to help divide a cell in two. the endomembrane system. Endo means inside, so this is the inside the membrane system. These are a bunch of interacting organelles and structures inside the membrane that help to get jobs done. They include different ones that we'll talk about, like the endoplasmic reticulum, or ER for short, and the Golgi bodies and vesicles. This is between the nucleus and the membrane, and one of the big jobs that happens there is packaging lipids and proteins as well. We also can recycle and dispose of molecules and particles there, and this will also include part of the nucleus, the envelope or membrane of the nucleus, and another structure called lysosomes. So starting with the nucleus so important, it's like the control center of the cell because that's where the cell's genetic material or DNA is. A nucleus also has other little areas, almost like nucleus within a nucleus. It's called the nucleolus. There may be one or more in there, and that's where ribosomes are made. Remember ribosomes? We talked about them earlier. They're the subunits made out of RNA, a type of RNA folded upon itself, like a bunch of fists that kind of congregate together like this, like two fists coming together, and they help to make proteins. So those ribosomes are actually put together inside the nucleus, inside the nucleolus. Then the nucleus has a double membrane, nuclear envelope, and there's a bunch of liquid or jelly-like fluid called nucleoplasm in there. The nuclear membrane can control the passage of different substances that are going in and out of the nucleus, and there's lots of pores for that, much like the plasma or cell membrane does. Chromatin, there's that word. So chromatin is basically DNA and proteins. It kind of reminds me of like spaghetti and meatballs, whereas the spaghetti is DNA and the meatballs are the protein. And so your DNA basically is just kind of floating around in there, all unorganized along with these proteins. While it's in that form, it's called chromatin. It's basically unorganized DNA. So during a regular cell life, that's what your DNA looks like. It's the chromatin form. But when it's ready to divide, when the cell is ready to divide, then it wraps around those meatballs or wraps around those proteins and becomes the visual structures that we're more familiar with seeing in pictures called chromosomes. So that's organized DNA, and that actually helps the cell handle the DNA a little bit better when it forms those chromosomes. So when you see chromosomes in a cell, you know that cell is getting ready to divide. Otherwise, when it's organized and looks like just spaghetti and meatballs, then we know the cell is not in its division cycle. Then there's also receptors and transporters that are on the nuclear envelope that are going to help things move across. And the outer bilayer of the double membrane then is continuous with something called the ER, the endoplasmic reticulum, which we'll look at next right here, the ER. It's like a bunch of passageways. It's classified as an organelle, meaning that it's a specific structure that has a specialized task or more than one task, and it has its own membrane. So basically it's these membrane passageways to move things through the cell, like kind of like the hallways of a cell or channels of a cell. All right, so along the ER you may find ribosomes. If that's the case, you call it rough ER. If you don't find ribosomes, then that's the smooth ER. All right, the rough ER, that's a big spot where proteins are made, or what we call polypeptide chains. That's before a protein is finished. And so we make proteins there, and then those little passageways, we can send those polypeptide chains through those little passageways. In the smooth ER, where there's no ribosomes, that's where your cell has enzymes that are assembling lipids that help to form the cell membrane, and also other substances are being broken down there. So this is what that looks like. There's the nucleus with the nucleolus. That's where the ribosomes are made inside here. We can see the chromatin. Doesn't look like chromosomes yet. It just kind of looks like spaghetti at this point. And then we have the outer membrane, which is continuous with the ER. So it's like you can go through this little pore and then enter a passageway right away in the ER, and you can just travel down the passageway. Then you don't have to step out into the cytoplasm and get wet, right? Then all these little red dots, those are the ribosomes, so that's all rough ER right there. In this picture, I don't see any smooth ER. Then there wouldn't be any ribosomes on that. So this picture doesn't have any smooth ER. So let's say a protein is made, and we should say a polypeptide chain, and that moves through these little passageways. Where's it going to go? Well, your answer is right here, the Golgi. The Golgi is like the UPS, or the shipping department of the cell. It finishes those polypeptide chains, folds them, gets them all made into the finished product called the protein, and then it puts a little vesicle around it and then can ship it to where it needs to go, just like a shipping department, like UPS or FedEx. So the Golgi body's job then is to modify those polypeptides and lipids, and it might need to attach little chemical groups like a phosphate group, and it can sort and package things and then bubble off into a vesicle and can send things into the cell or to the plasma membrane to be shipped out of the cell as well. So some places we see this called the Golgi body, sometimes they're called just Golgi, and sometimes Golgi apparatus. Anyway, they look a lot like the ER. So if I go to this picture here, this is the electron microscope, transmission electron micrograph, and this looks a lot like the ER, just a bunch of folded, kind of membranous passageways. That on the ends, though, you kind of see bubbles. That's because Golgi is really good at bubbling off vesicles with the finished products that it has just modified and packaged. So speaking of vesicles, those are small membrane enclosed sac-like vesicles that have different types. A lot of them can bud off of the Golgi, and they can transport things from one organelle to the next, or to the plasma membrane to go out of the cell or somewhere else. Other vesicles may have, if they're not carrying something specific, they may have maybe digestive enzymes in there, like lysosomes might, or peroxisomes. There could be more of a storage type of function, then we would call it a vacuole. And some cells have a big central vacuole, like a big, huge, basically storage membrane. But since it's for storage, they call it a vacuole. Like a plant cell has a big central vacuole. We usually don't see big central vacuoles in an animal cell, though. So lysosomes, we get our definitions here. Those have the enzymes and are good for digesting things. In fact, if you're like an amoeba, a single-celled organism who doesn't have a digestive system like you do, you rely on your lysosomes to digest things. So after an amoeba, a single-celled protist, engulfs what it wants to eat, like maybe some organic material or another protist or some bacteria, then it will bring it into the cell via a vesicle, and then it'll merge that with a lysosome, which carries the digestive enzymes, and that'll allow that single-celled organism to digest its food. And once it's digested, then diffusion can take place to take those nutrients into the cell itself. That whole process we'll get to a little bit more in our next section. It's called phagocytosis, and it's basically engulfing something if you're a single-celled organism, or it's how our very own white blood cells engulf things like pathogens within our own tissues. Peroxisomes are enzyme-filled vesicles that can break down things like amino acids, which make up proteins, fatty acids, which make up lipids and other toxic substances. Then we've got the vacuoles there again, those are for storage and stuff. I'll also mention the central vacuole in plants help to regulate water and help with kind of the pressure of the cell and holding that cell's shape. Here's a visualization of phagocytosis where we have a single-celled macrophage, which is a white blood cell engulfing a bacteria, and that brings it into a vesicle. So we can actually bring things into the cell without losing any of our very own cytoplasm, because this is going to close off right here, and we're not going to lose anything, and that vesicle then will be around what we brought in. There it is. Then we'll merge that with a lysosome containing the digestive enzymes, and we'll be able to digest that organism and make use of the nutrients. Here's a great picture going from the nucleus to the ER with the little ribosomes, little red things on there. And looks like we made maybe a polypeptide or a protein, and we're sending that here to the Golgi. Notice how the Golgi looks a lot like the ER, but it's just a little further out from the nucleus. And that Golgi then is going to finish that protein and modify it and package it, and maybe send it into the cell cytoplasm, or in this case in the picture, we are sending it in this part here to the plasma membrane, and we're exporting it out of the cell. Oh, and we have a picture up here of what the smooth ER looks like, where phospholipids are being made. It's almost like an assembly line. Ribosomes, these again are made in the nucleolus, in the nucleus of the cell. They're composed of the nucleic acid called RNA, 2 subunits actually, and then they're sent out of the nucleus to the ER, and some are free-floating in the cytoplasm, and then they are going to be in charge of protein synthesis, which will be another chapter down the line. The powerhouse of the cell, the mitochondria, the mighty mitochondria. It's a double-membraned organelle, produces ATP, adenosine triphosphate, which is a type of nucleic acid with just 1 adenine and 3 phosphate groups and a sugar. And that is basically our main fuel for our cells, one of the main fuels, ATP, made in the mitochondria. But in order to make that ATP, we need to have fuel to make it. We need sugar and other substances that we eat. So that's why you have to eat. You have to have sugar, glucose, other materials to be able to make this ATP in the mitochondria. Once you make the ATP, then the cells can use that for movement and other processes that require energy. So that's called aerobic respiration, when we're making ATP all the way through in the mitochondria. There's a theory that, well, those mitochondria, you know, they really are special when it comes to organelles inside a eukaryotic cell. They're kind of odd in that they have their own DNA. They have their own ribosomes. They make their own proteins. They can literally move on their own around the cell. They can reproduce on their own. And they really resemble a bacteria and are about the size of a bacteria as well. So there is a theory based on all of this evidence. This is very strong evidence that supports that the first eukaryotic cells engulfed prokaryotes but didn't digest them, and a type of prokaryote or bacteria that could make ATP. I mean, what a benefit that you have this inside of you if you're a cell trying to survive, and you've got this other cell inside of you that's much smaller, but it's making ATP, making energy for you. So you don't digest it and you keep it, and it's able to reproduce inside of you. And that's called endosymbiotic theory. Endo meaning inside, symbiotic meaning that there's a benefit for both of the organisms involved. Obviously, the main cell, the big cell, would benefit from the energy that was produced from this bacteria, could make ATP. And well, the bacteria that's living inside of the big cell then would have protection and food materials and a place to live. This is also true of one of our upcoming organelles called chloroplasts, that they're very similar, and again, they have their own DNA and ribosomes, they make their own proteins, they move around, they reproduce on their own, and they resemble bacteria. So endosymbiotic theory. So here is a transmission electron micrograph of a mitochondria inside of a cell. You can see it's double membrane. And you can even see how the membrane kind of folds in to the inside of the mitochondria. That basically just gives the mitochondria more surface area to be able to do more reactions to make ATP. Let's talk cell walls. Some organisms, you're a single-celled organism, may need a little extra support and protection, so they have a cell wall. And we've already talked about bacteria or other prokaryotes having cell walls made out of peptidoglycan. And you try to say it fast yourself. But plants have a cell wall made out of cellulose, which for us, that's actually fiber, something we can't digest. It's a carbohydrate that we don't have an enzyme to break down. So when we eat plants, we ingest their cell walls, which we can't digest ourselves. So that goes through us into the large intestine. It attracts water into our intestine and helps keep things moving through. So these cell walls can be not only beneficial to the organism that they're covering and protecting, but they're also beneficial to us when we eat them. So chloroplasts, we're going to find these structures in producers. So that would include plants and some protists like algae and some photosynthetic prokaryotes like blue-green algae or cyanobacteria. So they have kind of like the mitochondria, two outer membranes, and then a fluid inside, and then these stacks of disc-like structures that contain the green pigment chlorophyll, which is good at absorbing sunlight, which the sunlight then is used to power the making of sugar. There's a picture of our chloroplasts right there. There's the discs that I talked about that contain chlorophyll. And unless you're a producer, you don't have these in your cell. The extracellular matrix, extra means kind of the outside, and most cells of the multicelled organisms then would be surrounded by a fluid and many other substances like fibrous proteins, polysaccharides, and things like that. And even though it's just kind of the fluid outside of cells, it's very important because you're going to have things in there that are traveling to cells and will support cell signaling and keep things moist and support secretions and things like that coming from cells. So there's a nice picture of the extracellular matrix. the different things that may be in there, from carbohydrates and proteins. So this is the basis of our tissue organization and provides structural support out there as well. So here's some examples. Bone is mostly extracellular matrix composed of collagen, a fibrous protein, and some hardened mineral deposits. And blood clotting is also a role of extracellular matrix. So cells need to talk to one another as well. They need to communicate with one another. And that's often done by chemicals. Could also be done by touch as well. But how do you get chemicals from one cell to another? Well, you can have little junctions or tubes to travel between them. So plants use what are called plasmodesmata. Those are little cell junctions that can connect the cytoplasm of adjacent or neighboring plant cells. That allows different signaling molecules to flow quickly between the cells. And animals use what are called tight and gap junctions and desmosomes. So here's the definitions of those. So those tight junctions are basically arrays of fibrous proteins that can join epithelial cells together. It joins them very closely together to keep them from leaking. So a great example of where you would need that would be the epithelial or covering cells of the bladder. We obviously wouldn't want the urine and toxic wastes to leak out of the bladder into our body cavity. We want that to go out of the body. So thank goodness to these tight junctions that keep those cells very tightly kind of welded together. Desmosomes are like spot welds also that are between epithelial cells, and they keep the cells in kind of a sheet-like formation. And so places where we would need that would be like the skin, the heart, and the muscles, and we're only going to find those in animal cells. Gap junctions are very similar to the plant cell plasmodesmata, and they form little channels between cells. Basically, the plasma membranes are connected then between adjoining or neighboring cells, and there's a little tube in between that will allow stuff to flow between those cells, and that's called the gap junction. That allows cells to basically communicate and talk to one another through chemical signals and to share substances. And here's some pictures of those things, of these different types of connections between cells. The plasmodesmata in picture A between some plant cells. The tight junctions in B right here. Desmosomes in picture C right there. And the gap junctions in picture D. All right, so we finished our slides here, and when you're going through the book, then you're going to come to the next part, part 4 in chapter 3, which will start off talking about the cell membrane, but we're going to save that for next week. So we'll cover those 3 parts, the last 3 parts of chapter 3 for next week. Again, as you're looking at the end of this chapter, as I always encourage you to do, is to look for the questions that pertain to what we've just talked about in the first 3 parts. Question 1 and 2 there would apply. Question 3, that'll be next week for our art connection questions. And then the review questions here, look for the ones, again, pertaining to our first 3 parts. And the critical thinking questions as well, like question 15, 16, 17, and getting to, number 18, but maybe more so for next week, and 19 and 20 will be for next week. All right. Hope you enjoyed learning about cells, and we'll pick it up with the cell membrane and transport of materials next week.